Okay, we just have a few chapters left, and the next chapter that we are going to look at is trait inheritance. The first slide, where to go? The first slide is about Gregory Mendel. So he is considered the father of genetics, and because of his work, he discovered the genetics of inheritance and how things work. So he is a very, very famous man in biology. I found an article on nature.com, a very short article, not long at all, just some key information about Gregory Mendel, what his background was, and how he discovered the rule or the principles for inheritance. So read that short article, and I have a list of questions, very easy questions, that you can try to answer based on what you have learned, and I may have a couple questions on the test. Okay. So to kind of describe what his experiments were, actually you guys have already known most of this, but back then when we didn't know anything about the principles of genetics, we didn't even think about what genetics was. We didn't even know what DNA was, what RNA was. But Mendel did so many experiments, and because of his close observations and his very good record keeping, he made really good notes on his experiments. He finally figured out the principles of inheritance. Okay, so some of the information are actually on the slide. So he used a couple plants, but mainly peas. You guys like the green peas. Some of the frozen peas are really kind of sweet, right? When you heat it up in the microwave, it's really good. And I also like to put some in fried rice, and it makes it taste really good. So Mendel used the peas, and he did a bunch of crosses. So first of all, he did some crosses between the parents, which we call the P generation, right? P stands for parents. The two parents differed by one trait. So, a plant may have, not may, right? They definitely have multiple traits, right? Like the height, of the plant, what the flower color is, the shape of the leaves, right? Many traits. But he only focused on one trait first, and that's the color of the flower. So the two parent plants differ in the color flowers over here. One plant has violet or purple flowers, and the other one has white flowers. And now we already know that these two parents are pure, you can say pure, or they're homozygous for flower color. So the violet, the purple, is the dominant trait. So P for purple, and that's the dominant. And the little p is for white, and that's the recessive. So he used homozygous dominant as one parent, and then recessive for the other parent plant, and he crossed them. And he found that F1 offspring, so F1 means the next generation, all express the traits of one parent. And you know what the trait is, right? That's the purple flower. And do you know what the genotype is? If you have these two cross, and if they follow the inheritance rules, that's going to be big P, little p, right? And then this is a heterozygous dominant. Okay. All right. And now, if you cross the F1 generations, you take some plants from F1, cross them again, and you are going to find F2 generation, and that's a 3-to-1 ratio in purple flowers and white flowers. Okay, so purple to 1, that's 3-to-1. And now you already know, right? If you cross 2, oops, that should be big P and this little p. So just real quick. So the first 3 squares, 1, 2, 3, those are all purple, right? So you have 3 purples, and then you have just the last square, PP, that's 1, that's white. So the ratio is 3-to-1 purple to white. So he also noticed that there is a 3-to-1 ratio. I kind of want to point this out because it's so important. You probably noticed that the numbers here, 700, 220, it's approximately 3-to-1 ratio, not exactly, but about 3-to-1. And that's what you would observe in like a real experiment, because things can vary a little bit, right? The theoretical number is 3-to-1, but your observed value will be a little bit off, but very close to 3-to-1. And another thing I want to point out is that you probably notice how big these numbers are, right? So in total, that's almost 1,000 flowers. So he would, or 1,000 plants, right? So he would have to, observe so many plants and make notes for each plant. So he's done hours and hours and hours of work to figure this thing out. By examining large sample sizes, like I said, 1,000, and really just for this experiment, and he probably have performed many experiments because he showed that his crosses behaved reproducibly. So every time he did experiments, he did over 1,000 plants, he would observe similar results every time. Every time there's about 3-to-1 ratio for the F2 generation. The traits are inherited as independent events. He also noticed that observable traits, okay, like the purple in F1, right? The purple is the only thing that's observed in F1. But if you cross F1 again, in F2, you may have a purple and a white, but the dominant trait is purple. So he referred to observable traits as dominant, and then the non-expressed, or the one that could be masked by the dominant trait, as recessive. So you guys have already known that. So it's just some real quick some terms to kind of help you kind of refresh your memory. So we talked about phenotype and genotype before, and we also had some questions on the lab test, I believe, on the differences between genotype and phenotype. So you know that one is going to be the observable trait, and that's your physical appearance, right? So what you look like. So if you look at the picture down here, this picture shows you two people with different hairlines. So the first one, you already know who that is, very famous. So you can see the hairline it's a little bit the middle kind of sticks out a little bit, so we call that widow's peak. And then the other guy, I really don't know who he is. I have asked many people, but nobody knew who he was. I just accidentally found this comparison online, no idea who the second man is. And you can see that his hairline is flat, right? So that's called a straight hairline. So what kind of hairline you have, whether it's a widow's peak or straight hairline, that's the phenotype. And genotype is, so his widow's peak is actually the dominant trait. So he could be homozygous dominant or heterozygous dominant. We don't know. We have to look at his genes in order to figure that out. Or we can, use his parents' genotype to kind of figure out what he has. And then the straight hairline is recessive. So we know the second guy is definitely recessive, right? And that goes, that's definitely saying that this is homozygous recessive, right? So a lot of times we just not say the homozygous, you can just say it's recessive. So this is a genotype, and what they actually look like, that's the phenotype. Alleles, so alternative forms of a gene. So in this hairline example, the big W and the little w, those are different forms of a gene, right? One codes for widow's peak and the other one codes for straight hairline. So we use letters to indicate different versions of a gene. So those are alleles. Locus, the point on homologous chromosomes where alleles are found. So basically, this is the location on the chromosome where you can find a gene. So remember, the homologous chromosomes, they carry the same genes, right? But could be different versions because one chromosome comes from your father, one chromosome comes from your mother. All right, so we have really figured out the different locations on the chromosome, and we have already known the locations for most of the genes. So let's say the hairline gene is located on this particular chromosome, and it's on this section right here. So this could be a let's say big W, it's a little w, this location. Now we can use letters and numbers to indicate that location. So that's going to be the locus for the gene that controls your hairline. You already know dominant gene, recessive gene, stronger, weaker. But know that this is not always the case. We are going to talk about some non-Mendelian genetics because they do not follow the rules for Mendelian genetics. And in those cases, sometimes you don't have a dominant or a recessive gene. Next pair of terms, heterozygous, homozygous, you already know that, right? If you have a different versions of genes, like down here for hairline here, so big W, little w, that will be heterozygous because that's two different versions. Homozygous will be homozygous dominant or recessive. Okay. Now, Punnett square, you've already know how to do the Punnett square, so I'm just going to skip that. Laws of inheritance. It's a lot of reading, so make sure you read this slide and get familiar with some of the terms and ways we use to describe laws of inheritance. So Mendel postulated that genes, which kind of code for different characteristics, are inherited as pairs of alleles that behave in a dominant recessive pattern. Again, this statement was true back then, right? But now we have discovered some genes, some characteristics are not inherited in the dominant recessive pattern. So basically, you inherit two chromosomes, again, one from the father, one from the mother, and there are pairs of alleles on those on that pair of chromosomes. So these two alleles, again, what you inherit from your father and your mother, control what characteristics you are going to have. Each gamete is equally likely to receive either one of the two alleles present in a diploid individual. So you have to kind of go back a little bit. So this is the chromosome, let's say from your father, and this is chromosome, the pair of homologous chromosomes from your mother. All right. Now, when they make gametes, you know what, just to make it a little bit clear, so let's say your father carries two different alleles, two different versions of a particular gene. So we're going to call it 1 and 2. And it's the same thing for your mother. Let's say your mother also carries two different versions. So when sperm cells are made, right, when your father's body produces sperm cells, it's called spermatogenesis, right? And the original cell, germline cell, spermatogonia, is going to, go through meiosis, right? And eventually you are going to have a gamete. So I'm going to say gamete. Each gamete equally likely to receive either one of the two alleles. So this gamete may carry number 1, allele version or gene version number 1, and that's going to be sperm, right? Let's say sperm. And then the other sperm may also carry the other allele, right? Because remember, it's just kind of separated. So the sperm has equal chance of carrying either 1 or 2, right? For the same gene. And same thing for a mother. So let's say this is the sperm. Same thing for a mother, right? So an egg could carry gene version 1 or gene version 2. And then they are going to fertilize, right? So fertilize everything is by chance. So this is how you can make the Punnett square. So this is based on equal chance probability, which leads to the Punnett square, right? If you put, so this is the sperm that carries gene version number 1, and this is the sperm that carries gene number 2. And same thing for here, mother 1 and 2. And then you do different combinations, right? So if this sperm by chance fertilizes this egg, you're going to have a 1, 1, and then do the other combination 1, 2, 1, 2, 1, 2. Oops, 2, 2, sorry. Okay. So that's pretty much the basis for inheritance, and also the basis for the Punnett square. Okay. In addition, genes are sorted into gametes independently of one another. Alleles are generally not likely to segregate into a gamete with particular allele of another gene. So that's pretty straightforward. So I'm gonna skip that. And then the next one, okay, this is something that you haven't learned yet from the lab. This is a di-hybrid cross. And you know what di means, right? Di means two. So far we have only done mono hybrid. So mono means one, so it means that we have only looked at the inheritance for one trait. So it's either the color of the flower or the hairline. But now we're going to introduce a second trait and see how everything works. Okay, so here is what it looks like. Now you notice that the Punnett square is a little bit bigger, right? Instead of 4 squares, you're going to have 16 squares. And again, that's because you have to figure out all the possible combinations of alleles that a gamete can carry. So instead of a one allele, Y or R, because now you're looking at 2 traits at the same time, so you're going to have Y, R together. And now you're going to have 4 possible gametes, or gametes that may carry one of the 4 possible genotypes or different combinations of alleles. All right, so let's look at this Punnett square. Here, the YY, that's for color of seeds. So yellow means, sorry, I already told you. So Y is the dominant trait, so that's yellow. Okay. And then the little y is going to be the recessive, which is green. Right? Okay. And then you probably notice there's another letter, right? So the big R is for smooth. So when you look at seeds, sometimes it's very smooth, sometimes it's very wrinkled, right? And then the little r is the recessive, which is the wrinkles on the seeds. All right, now you can kind of imagine how much work Mendel would have to do to figure all this out with the 2 traits. All right, so let's say this is the genotype for the 2 parents. Again, remember, Mendel always started with quote unquote pure or homozygous, right? So the seeds are homozygous. They have the same 2 alleles for each gene. So the parent trait is going to be yellow and smooth. Okay, so everything is homozygous dominant. And then the second trait is green and wrinkled. All right, so again, this is also dominant, or sorry, not dominant, but also homozygous, but it's very recessive. And then he crossed the 2, and this is the F1. And you know F1 is heterozygous dominant, right? So when we look at this is a heterozygous for color, and this is a heterozygous for the texture of the seeds. And then he is going to cross in between F1. So he would just let the plant self-pollinate. So F1 pollinates F1. And this is the all possible gametes from heterozygous parent. Okay? So what you can do is to write out the genotype, and you can do different combinations. So what if a gamete inherits a Y, the big Y, I'm just going to say big Y, small or little y. So what if a gamete inherits big Y and the big R? So that's the first possibility. Okay. The second possibility is the gamete inherits a little y but big R. Okay. So that's the first possibility. That's the second possibility. So that's the second one. The third possibility is you inherit a big Y but a little r. Okay, so that's the third. And the last one, little y, little r. That's the fourth possibility. Okay, over here. And then it's the same thing for the other parent, right? So the parent can generate these 4 possible gametes. And then let's make different combinations with these 4 different possibilities. So if you have this gamete, fertilizing this gamete, then this is what you have, the genotype for the seeds. Okay? And the same thing here. So you just kind of write out genotypes for all possibilities, and this is what you see. Let me explain the phenotype they assign to each seed. So basically, they have already shown you. So over here, that means yellow and smooth, right? That's what you see. The seed is yellow and smooth. And over here, little y, that means green, right? So the seed is green, and big R, big R, that means the seed is smooth, right? So green, smooth. But if you come here, what do you have here? This is heterozygous for yellow, right? The dominant trait, so it's yellow. But over here, little r, that's a wrinkled seed. So that's what the seed looks like. And same thing here, So you got both recessive traits, green and wrinkled. Okay, and then when you count the different phenotype, so follow me. What phenotype is this one? Yellow and smooth, right? Okay, so I'm just smooth, and this is green and smooth. And the next one is yellow, wrinkled, and this one is green, wrinkled. So if you go through each square, you will only count for different phenotypes, and these are the correct phenotypes. And when you count the number of squares, it's 9 to 3 to 3 to 1. So you have to memorize this ratio, just kind of like how you memorize the 3 to 1 ratio when we talk about monohybrid. This is dihybrid, and the ratio for different phenotypes in the offspring is going to be 9 to 3 to 3 to 1. So remember this, 9 is going to be the seeds that have both the dominant traits. So both dominant. And then the 3, that means they only have one dominant trait. One dominant. So whether it's yellow or whether it's smooth. And this one, yellow, that's dominant, but wrinkled, that's recessive, right? So again, this is only just one dominant trait. And then for the ratio 1, that means you have 2 recessive, right? Both traits are recessive. So it's actually pretty easy to remember. Complicating the law of independent assortment. So this is what we kind of refers to some exceptions, which we discovered after the Mendel genetics. So we have noticed that sometimes the chromosomes that carry genes, right, they don't just, separate and be distributed independently into the gametes. Sometimes exceptions do occur. Alright, so here's what may happen. When genes are located in close proximity on the same chromosome, their alleles tend to be inherited together. So if you are talking about genes, again, a single chromosome may carry more than 1,000 genes. So here is an example. All right, so this is one pair of chromosome. This is another pair for comparison. If the two genes are quite far from each other, so let's say this gene 1 and gene 2, and then this is just the same gene but on the other chromosome, so I'm not gonna write it out. So if the genes are very far from each other on the chromosome, then they follow the rules for independent assortment. They don't affect each other. Okay, but if two genes are very close to each other on the chromosome, then they may be inherited together. So when you look at the traits, the two traits inherited or coded by these two genes, G1 and G2, then they may not follow the independent assortment that we see here. So instead of a 9 to 3 to 3 to 1 ratio, you may see a very, very different ratio because these two genes, they tend to link with each other when they're distributed into gametes. Okay, so they start to kind of they like to stay with each other. They don't like to be separated based on the rule of independent assortment. Okay. And we also have recombinations. So recombinations is really what crossing over is about. Okay. So random event occurring anywhere on the chromosome. So genes that are far apart on the same chromosome are likely to still assort independently because of recombination events that occurred in the intervening chromosomal space. So it sounds really kind of complicated, but just think of this. Remember when the homologous chromosomes pair up in prophase 1, you may have exchange, right, in section of the chromosome arm. So the genes may be switched, right? So instead of big C, now you have the little c. And then same thing here. So this could totally mess up the independent or assortment, the rules that we just talked about, things that we can use the Punnett square to solve. So the crossing over will also undermine that rule and create some exceptions. Okay. Here, just for fun, you can look at what traits you have, and usually these traits can still be considered dominant and recessive. But when you look at the ratios in the population, they don't strictly follow 3-1, and that's really because some of the things that we have mentioned, right? Some genes may be very closely related, not related, but they are located in proximity on the same chromosome, so they may be linked when the chromosomes are separated and end up in different gametes. So that's one thing, and also crossing over, right? And also, some traits may be determined by multiple genes instead of just one gene.